You will need these items from the AutoPlasmid MMG kit for your sample preparation along with your thawed cell pellet. First, add the R Nase A vial contents to the Resuspension Buffer bottle with the pink cap. Twist the lid back on, and invert the resuspension bottle 2 to 3 times to mix the contents. Then, add the resuspension mix to the clear cell pellet bottle. Twist the cap back onto the cell pellet bottle and shake for 15 to 30 seconds to mix contents. The resuspension beads will aid in dislodging the cell pellet. Now, add the contents of the Lysis Buffer bottle with the orange cap to the cell pellet mixture. Twist the cap back onto the cell pellet bottle and slowly invert the bottle 10 to 30 times. The solution will turn blue when it's fully mixed, but be careful and do not exceed 5 minutes during lysis. Then, add the contents from the Precipitation Buffer bottle with the green cap to the blue mixture in the cell pellet bottle. Twist the cap back on tightly and gently invert the bottle 10 to 30 times. The solution will turn clear when it is fully mixed, and the cell debris will be visible. The plasmid DNA remains in solution while the cell debris is precipitated. Finally, pour the bottle contents into the sample filter plate, and your sample prep is complete. Initialize instrument software to start the run. After the run has finished, collect the purified plasmid DNA.